So we're going to go over membrane proteins and some of the types of transport. Also, if you want to download the notes that I used to make this video and this image itself, I have it in the captions box below. So let's go over intrinsic or integral membrane proteins, and that's this guy right here. And so integral membrane proteins, as they sound, they're gonna span across the lipid bilayer. So you're gonna see that it's gonna go through the entire lipid bilayer, and it's gonna span the length of the phospholipid bilayer. It's going to have membrane spanning regions of alpha helices with hydrophilic amino acids. And those are the side chains. Because remember, these heads are all hydrophilic, so you want the side chains to be hydrophilic as well. And these side chains are going to be lying within the alpha helices. And some examples associated with this. So the examples that I can give you are the sodium potassium ATPase, glucose transporters, and the ion water channels. And these are specific towards the integral proteins. And so this will typically see axial rotation and rapid lateral diffusion, meaning that you're going to have some rotation of this integral membrane protein, and you'll have some lateral diffusion going this way. The proteins themselves, they don't move across to the other half of the bilayer, so you're not going to see kind of this motion in terms of the integral membrane protein. So it's basically not going to go this way, but you may see some lateral movement this way. So once again, movement can occur across the lipid, or transitioning through the bilayer, but you're not going to see a flip. Now when we're looking at extrinsic proteins, which is this guy right here, they aren't going to penetrate the lipid bilayer itself. Instead, they can either be on the extracellular or the intracellular side. And typically, the attachment that you see here, I put a space there to symbolize that it's going to be a weak ionic interaction. And so what that means is it's allowed to sit here, but it's not going to... It has the freedom to move if it wants to. So the interaction is weak enough to allow for this protein to sit here, send the signal inside, but it doesn't have to stay fixed the whole time, unlike the integral, which is pretty much fixed along the lipid bilayer, meaning that you're not going to see this entire unit leave at any point. So then if we look at the extracellular surface, and specific with carbohydrates, just remember that it's 10% glycolipid. There's also glycoproteins and monosaccharides. And the monosaccharides, in terms of the carbohydrates, are important for cell recognition and determining receptor specificity. So what are the types of transport that are typically seen along the lipid bilayer? And so there are a couple different types. There's active and passive transport. So we have, for transport, we have active and we have passive. And the difference between these two types is the energy. So active is going to require energy, while passive does not. And the types of transport in these groups are, you have simple diffusion, and then you have facilitated. While in active, you'll have primary and secondary transports. And these two are going to be carrier dependent. So now if we think about passive, typically what passive is going to mean, that it's going to be going with the concentration gradient. So it's going to go down the concentration gradient. So if you think of a hill, so if we think of this hill, right? The passive side is going to be this side right here. So if we have a stone up here and we're rolling it down, that's considered passive. Now if we're looking at active, active is kind of the opposite. So we have that same stone and we're rolling it up the hill. This is going to require energy. Same thing as if you're rolling a big boulder up a hill, that's going to require a lot of energy to do that. But the moment it hits the max and you just tap it, it's going to go on its own down the hill. So that's an easy way to remember passive versus active transport. So typically passive is going to go down an electrochemical gradient. While active transport is a chemical gradient. So that's another thing we should probably write in the table. So this is chemical, and passive would be electrochemical. And then if we think about the types, so with simple diffusion, what that really means is, this is going down its concentration gradient. It means that ions are going to be able to pass through without any problems. So if we have, as we said before, the high concentration of potassium inside the cell, actually this is a bad example. So we have some ion, we're going to call it. It's going to easily be able to go out of the cell because it's going with the flow of the concentration of all those ions associated. Facilitated diffusion, on the other hand, it's going to need a helper. So it's basically going to leave the cell with the aid of another ion. Now if we look at something called carrier-mediated transport, and don't worry, we're going to get into these in the next video. If we look at carrier-mediated, what that means is, this is kind of the idea of facilitated diffusion. In carrier-mediated, we're going to have some channel where we have this ion, and then we'll have a carrier kind of help. So that's what we're talking about right here, the carrier-dependent groups. And the carrier-dependent groups, as they're helping to come out of the cell, what's happening is there's going to be a point where there isn't enough carriers to help take out the ion, or take out molecules of glucose or whatever it's going to take out. So what will happen is it has the ability to get saturated. So if we were to look at the graph of a carrier mediated, we are going to see at some point it's going to plateau, and that's usually known as Tmax. Also, specificity is important. So if we're thinking of the specific ion, we want to make sure that whatever we're carrying across, it's what we want. So if we're taking sodium across, we're taking sodium outside or bringing it in, we want to make sure that the carrier is only going to take sodium and not potassium or not chloride or any other ion, because we want to make sure that the intracellular space has the correct concentration of ions or the correct composition. So that's the third thing that's important with carrier mediated is composition. You want to make sure your solutes are the same that's needed by the system on both sides. So now the question becomes, what's the difference between some of these methods? So if we look at ion channels, which we've kind of been discussing, versus facilitated diffusion, well, these ion channels that are involved in simple diffusion, as you said, these are electrochemical. While facilitated is chemical. The difference is this ion channel, you want to make sure that you're controlling what ions go in. So on for the most part, it's off. And then whenever there is a charge difference, this can now go into the on switch. While in facilitated, it can't be turned off. It's always going on, but it needs to have that help. The difference between these two, also in terms of speed, this one is super fast, while this is moderately fast. The ion channels are typically involved in electron signaling and secretory processes. So this is electrical signaling, plus secretion. And this is more for transport of sugars, amino acids, basically your bigger stuff that you're going to be able to transport. This is, as I said, ions, so it's small things. And if you think about it, if you're sending something small, like a sodium ion, it's going to be a lot faster than if you're sending something big. So if we think about the lipid bilayer itself, if we want to send something across it, we want to make sure that whatever we're sending, if we need it there fast, we want to make sure that it's small and then it's not going to get impacted by the layer. So basically we want to make sure it's lipid soluble. Because if it's not lipid soluble, it's not going to be able to pass here with ease. If it's a larger molecule, it's going to be, so if we have something big and we're trying to make it across, it's going to be very difficult to go across. So we want to make sure that it's a small ion, it's lipid soluble, and it can go through. Also in terms of acids and bases, so if we have HA, which is our acid, it can go through, but the moment we have, like say H3O+, it has this charge. This charged ion right here, or this charge that is carrying on this acid, is not going to allow it to go through. Because if we think of these head groups versus the tail groups, that charge is going to be attracted by this, and it's not going to allow it to pass through. But if we have, say, water, it can easily pass through the bilayer. I hope that made sense. If you have any questions or comments, you can leave them below. If you like the video, give it a thumbs up. And if you like the channel, please subscribe.